So let's take a look at some horrible diseases. Polio, measles, hepatitis, smallpox, chickenpox, monkeypox. Do you know what all of these have in common? Well, the title kind of gives it away. Right. These are all diseases caused by viruses. And coincidentally, these are actually all diseases that we now have vaccines for. So, what are viruses? How is it different than bacteria? How do we treat or cure viral infections? That's what we're going to look at in this video. Let's get started. So in addition to some of those viruses that I mentioned early on, one of the biggest pandemics of all time was the 1918 pandemic. You may have heard of it as the Spanish flu. That's actually a little bit of a misnomer because it actually didn't originate in Spain, like some people suggested. The Spanish flu and all flus are actually viruses. This particular flu had about 17 to 50 million deaths worldwide, right? And this was a really particularly bad flu, so much that our modern day vaccines actually have a little bit of protection against this particular flu strain. So what is it about viruses? Well, let's start by giving you a sense of size. So if you think about, eukaryotic cells, right? We can't barely see those with our eye. We could see like some fungus, some mycelia of a fungus. We could probably see those with our eye. Most eukaryotic cells, that's the cells of humans and animals, we can't see those most of the time. We need to use a microscope. Yeast, smaller than that. Bacteria, even smaller than that. Mycoplasma, right? Some of the very smallest of these are smaller. And then viruses are just tiny, 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 right? Viruses are not cells. If you look at these other things, right, these are cells. They have complete parts of life in them. A virus is not technically a living thing. It's just sort of, some people say it's a piece of DNA wrapped in protein, and it's basically just some bad news. Okay, so to see these viruses, we really have to use like electron microscopes to see them. So what is a virus? Is it alive? That's what I just mentioned. Basically, it's DNA or RNA, so we have some genetic material wrapped in a protein coat, right? A piece of bad news wrapped in some protein. Viruses are not cells. They don't have all the machinery of cells. So there's lots of different types of viruses, whether we're talking about a bacteriophage that infects bacteria, whether we're talking about different types of flu or even the SARS-CoV-2 that caused COVID. There's lots of variety. So what is it then? Well, some people like to label a virus as a parasite. Think of a parasite. What does it need? Well, it needs a host. So a virus can technically be considered a parasite because it needs the host machinery. Because viruses don't have enzymes for metabolism, right? They can't break down food. And they don't have ribosomes to make proteins. So where do they get those things? Oh, your juicy cells, right? Your cells have all the machinery they need. So these bacteria are parasites to your cells, and they use your machinery to replicate and spread. So I got a little example here of a machine, right? Basically, you are a machine. Your machinery is what the virus wants to use to survive. Okay, so we've talked about what a virus is, what it is not. Now let's show you a little bit of variation. So variation in viruses show that basically viruses are just these package of genes that are in transit from one host cell to another. And here's one of the scariest things, but one of the most sort of awe-inspiring things about viruses. Viruses don't travel. We, or animals or something else, carry the viruses around, right? They don't have little feet. They're not traveling. We actually take them to other hosts, which is really even more sad to think about. So here's a lot of viruses, lots of variation. I want to point out a few things to you. Notice some viruses are super, super tiny. A lot of the parvoviruses, the hep viruses, A, B, and C, right, that cause hepatitis. The human papillomavirus, very small. And then you get up to some bigger viruses. The Ebola virus is actually quite a bit bigger. Now, we're still pretty small compared to everything else, but it's one of the bigger viruses. The herpes viruses are pretty big. But what you're probably noticing, and as you look through all of these, right, basically what you have is you've got some genetic material in the center somewhere, right, some genetic material somewhere, surrounded by some proteins. Then you've probably got these little things on the outside. Some of these you might call a spike protein. These allow the virus to latch onto cells and get in. But we've got a lot of different variety here in these cells. But basically they all consist of the same thing. Some genes, DNA and RNA, that are packaged with some protein and trying to get from one host to another. All right, so speaking of the genetics, speaking of those genomes in the virus, there's lots of variety in these viral genomes. So you may remember from watching my other videos that humans have DNA, which codes for messenger RNA. And if you look at bacteria, if you look at plants, right, they all have DNA. It may look a little different. It may have round chromosomes versus linear chromosomes, different lengths of chromosomes. But we basically, most living things, have DNA. Now, viruses, they don't necessarily always have DNA. So let me give you some examples. Smallpox, right, a particularly nasty one, also related to monkeypox, actually has double-stranded DNA, which codes for messenger RNA, and then goes through the process. Another parvovirus has single-stranded DNA. But then if you go down here into some of these others, some of these rotaviruses, and then the coronaviruses, right, which caused COVID, those are actually RNA. Some are single-stranded RNA viruses. Some are double-stranded RNA viruses. Some serve as the primary RNA target, which helps messenger RNA be made. That's why you see these positive versus negative strands. So if you had a positive strand of RNA, that has to first be converted to the negative template strand, which will then, of course, be used to make messenger RNA. So you've got lots of variety here. And then HIV, which is even crazier, which is a retrovirus, and I'm going to talk about that more in a moment. That is RNA, which then goes to DNA, RNA, which goes to double-stranded RNA, which then, as a retrovirus uses, turns it into DNA, which then takes it, right? These are crazy. Bottom line, viruses have lots of different types of genomes. But what's common is that they have either DNA or RNA. Okay, so we talked about what's inside the virus, DNA, RNA. What about what's outside the virus? Well, the virus has a protein coat, which we call the capsid. That capsid is a crystal-like protein shell with a few different types of proteins, but they're mostly the same copies of the same protein. So here you see the genetic material, this red stuff inside, but the capsid, all these proteins, are what are on the outside. So you can see our capsid coat, we got our genetic material inside. Here's one of those subunits of the protein. So if you could just look at one, you'd notice this is what one looks at. But when they're all together, they form this protective coating around the virus. Okay, where can a virus live? We talked about the idea that they need a host. Well, some viruses have a broad host range and some have a narrow. Okay, well, what's a host range? Well, believe it or not, some viruses can infect many different animals, like rabies has a broad host range, can affect all mammals. So you think of dogs and humans can get rabies. Some are narrow. The cold virus only likes human respiratory tract. HIV only likes specific white blood cells. So while these viruses have different ranges, they like to specialize very often. So if we have a bacteriophage, right, a phage, which infects a bacteria, right? That's a simple narrow host. But then you have something like rabies affecting both of these people, both the male human and the dog have rabies in that, because rabies seems to affect all mammals. Okay. Then, again, some are more narrow in their scope. This specific one here is showing you HIV, how it only binds to certain white blood cells, the helper T cells, which are actually pretty important to our immune system. All right, so what about some emerging viruses? So sometimes a virus will jump host, right? So I was just saying some viruses have a broad host range or a narrow host range, like they're only in birds or they're only in bats. But sometimes we have a virus that jumps host. So we call this zoonosis, right? Where a pathogen jumps from an animal to a human. This is most likely, we think, what caused the SARS-CoV-2 shift, right? We think probably, maybe from a bat, although there's some evidence to say it possibly could have been a leak or escaped from a lab. I don't know that we'll ever know the answer to that, but we do know that pathogens can jump from animal to human. So here's some that I'm a little worried about. I think these might be emerging viruses, right? Ebola, it's been all over the news. SARS, we've just dealt with a pandemic from that. Then we need to watch these bird flus and swine flus. Those are some of the ones I'm worried about really getting into the human population and causing one of our next pandemics. Zika, one you don't necessarily hear much about, but it has some really nasty effects, especially if you're a mother carrying a child. It can cause some what we call microcephaly, or really small heads on a baby if the mom had Zika. And then, as I mentioned, some swine flu that we would see in some of our pigs or swine populations. Lots of really terrible symptoms from some of these influenzas. We need to watch those and be careful. Maybe now start to develop some vaccines for some of these things in case they really do jump host. All right, so let's talk about how a virus lives. What's its life cycle? Well, I told you it needs a host cell. All right, so all of this is your host cell. Okay, let's pretend it's your cell. So what has to happen is, here's the virus. It has to find some receptor to bind to so that the cell lets it in. In COVID, it happens to be something called an ACE2 receptor that tricks the cell, and then the cell says, oh, okay, come on in. So the virus comes in. When it comes in, it releases its viral RNA. Well, what then happens when the body finds, in this case, it also has some reverse transcription, so it went from viral RNA to DNA. What happens when you've got DNA? Well, it goes in the nucleus. What does the nucleus do? It might add that DNA. It might integrate it into some of your own DNA, transcribe it back to RNA, and then we know what happens from then. Transcription goes over to the ribosome, gets translated to make the proteins, that protein coat. Then it assembles itself. So once again, we've got this viral material, this, in this case, viral RNA, surrounded by the proteins that your cells made. Your cells made new virus. Then those viruses escape, they bud out of the cell, release, and go over to other cells. So this is pretty nasty, right? It's what viruses do. They basically take over your cell and use your machinery to replicate. So, viruses have highly efficient replicative capabilities, meaning they can replicate, that allow for rapid evolution. So if you were looking, as we went through this COVID pandemic over the past several years, you kept probably noticing, oh, there's this new strain. Oh, now we're to Omicron. Oh, we had Delta. Now we're to Omicron. Why did we have so many strains? Well, what happened was, because we didn't get rid of it early, didn't wipe it out, everyone didn't get a vaccine early on, it allowed time for evolution to occur. So those viruses, because they replicate so much, they evolve very quickly. And what happens is, as viruses evolve, basically they're replicating themselves. But as we've learned in different videos, when your cells make copies of DNA, RNA, they're prone to mutations. Well, since viruses are replicating so much, they seem to be extra prone to mutations, especially if it's one of the RNA viruses. So these mutations occur throughout this host pathway, meaning that more and more viruses are going to mutate more and more. So if you think about, I've got an example of some DNA here, right? One of the things that viruses also can do is combine or recombine information in the host cell that they infect. So for example, related viruses might combine information into a new gene into a cell. So you take some from this part of virus, some from this, then they recombine in your host cell, and then you've got a really bad gene perhaps, or one that's particularly good for the virus, particularly bad for us. Okay, so I mentioned early on this idea of phages. Phages are basically just viruses that infect bacteria, right? So we don't have to worry about this little thing infecting us, because these evolved to infect bacteria. So you've got this 20-sided capsid head, and you've got the DNA, the genetic material, inside. So what happens is, you can see the bacteriophage lands on your bacteria, so this would be a bacteria, lands here. Notice that all this genetic material, DNA, is injected into that bacteria. Then the bacteria thinks it's its own DNA and will make more bacteriophages. Okay? So bacteriophages are a particular type of virus that infect bacteria. So what are the life cycles of those bacteria? I want to bring up two for you. There's one life cycle called the lytic cycle and one called the lysogenic. If you break down the words, they really make sense. If you think of a lytic cycle, like something's going to break open and rupture. So here's an example of the lytic cycle here. One, you get these phages, they land on a bacteria, they inject their genetic material into the bacteria, then the bacteria takes that genetic material, right, transcribes it, translates it, makes new viruses. Those viruses assemble, rupture the cell as they leave. That's why we call it the lytic cycle. Another, I think a little more interesting step is the lysogenic. Think about this, lysing a gene. This is when the viral DNA gets incorporated into the bacterial DNA. So for example, if you look here, we go to first step again, so that's the same. Bacteriophage injects its DNA. Here's the DNA. Well, over time in the lysogenic cycle, notice how this DNA has become part of the bacteria's DNA. Well, what do bacteria do? They divide, right? So the bacteria divide several times, cell divide over and over again. So then basically what they've done is this virus has just been growing inside these bacteria over and over again until it might come back here and then go through the lytic cycle and break out. Okay? So, crazy little things, these bacteriophages. Well, what have bacteria done? Well, they actually, because they were tired of being, I guess they were tired, I'm anthropomorphizing bacteria. They got tired of these bacteriophages injecting this DNA, so they evolved some restriction enzymes. Restriction enzymes find particular places in foreign DNA and cut it up, right? So this enzyme, one particular enzyme, maybe it's in E. coli, right? We might call it EcoR1. This particular enzyme looks for a pattern that it has learned from the viruses exists. So it looks for this pattern here. Once it finds that pattern, it's like little scissors and cuts it up. So then you have two pieces of that material, of that genetic material, so that that viral DNA can't cause as much harm. Now, why do I bring this up? Well, because we actually use restriction enzyme. We use them in biotech procedures, right? So later on in my video about biotech, you'll see that we use restriction enzymes if we want to cut up DNA for some other purpose, okay? So again, these bacteria produce these restriction enzymes that recognize cut up foreign DNA, which we have actually learned to harness as humans and use in several biotech mechanisms. All right, so as we start to sort of wrap things up, I want to talk about a particular set of viruses, RNA viruses. RNA viruses in particular, because they lack replication error checking, right? You may remember DNA has the enzymes which go back and do a little check to make sure the DNA was correctly made. Remember, when we're making RNA, there is no check. So because these RNA viruses don't have the checking mechanisms, they have high rates of mutation. So they're constantly mutating, constantly evolving. So you may remember the basic flow. I've been talking about it, or I've alluded to it many times throughout the video. DNA goes to RNA through transcription. RNA makes the proteins through translation. Well, one particular virus which is really nasty are the types of retroviruses. Retro, think reverse. So they have RNA that they use reverse transcriptase enzyme to make DNA. All right, so the virus comes in with the RNA. It uses reverse transcription, because remember, transcription is normally DNA to RNA, but if it's reverse, it would be RNA to DNA. To make DNA so that then can go through the normal pathway of DNA to RNA to protein. This polymerase is actually transcribing the DNA into the messenger RNA. Now, there's a particular RNA virus, which is a retrovirus, which you've probably heard of, right? It is HIV. So that's what I want to sort of talk about here for just a moment, because this is such an important virus. HIV is a retrovirus. So let's see exactly what it does. HIV stands for human immunodeficiency virus, HIV. This is the virus, HIV. So HIV, the virus, causes AIDS, which is acquired immunodeficiency syndrome. This is an opportunistic disease which really lowers your body's immune reactions, right? Because HIV attacks helper T cells, your body can't fight off other infections. So HIV is the virus, AIDS is the syndrome. So one would never say, oh, they caught AIDS. No, they didn't catch AIDS. They caught a virus which causes the syndrome AIDS, okay? So here is what HIV looks like. You can see it's got its inside the virus. It has got its RNA, single-stranded RNA, but it also has that nasty reverse transcriptase, that enzyme which is going to take this RNA and convert it to DNA. So let me show you how that works in a cell. So here is HIV. HIV binds to a particular receptor, CD4 receptor. CCR5 is there too, which allows the HIV to enter specifically these white blood cells. Once it gets in, that viral RNA, now with the help of the enzyme, right, reverse transcriptase, takes this viral RNA and makes it into DNA. Well, then we know what happens. Same old story. DNA in the nucleus integrates itself into those white blood cell nuclei, DNA. Then what happens? DNA is transcribed into RNA. RNA goes out to the ribosome, is translated into what? More viral RNA, more protein, so that the HIV can assemble back, bud off, and go and infect another cell. Okay, so HIV in particular is a pretty nasty virus for many reasons, one of which it's an RNA virus and it mutates a lot. Second of which, it hurts your body's immune system, so you really don't have a chance of even getting rid of it unless you take quite a bit of antiretroviral drugs. All right, last slide, I want to talk to you about the last class of viruses, which are viruses which we know can cause cancer. There are several viruses which are now appearing to cause certain human cancers. Hep B, we know, is linked to liver cancer, right? So there is a vaccine, by the way, for hepatitis B you could get. Epstein-Barr, it seems we're finding more and more that this Epstein-Barr virus, which you've probably heard of as mono, if you get mono, you've got the Epstein-Barr virus. We're now thinking that this is linked to some lymphomas and perhaps even multiple sclerosis. Papillomaviruses like HPV, linked to cervical cancer in women. Ladies, that's why it's important especially to consider getting that Gardasil vaccine, because it protects against HPV, or at least having that discussion with your doctor. And then HTLV1 retrovirus is actually linked to adult leukemia. So once again, these viruses tend to wreak havoc, whether the virus is causing HIV, whether it's causing a horrible flu pandemic or the COVID pandemic, or, as we see here, causing problems that lead to ultimately cancer. These viruses can be nasty little things. That's why I think more and more studies need to be done on viruses, more and more studies about vaccines to sort of help us have a fighting chance against these things which are mutating so rapidly.